Well, welcome everyone to the July installment of the IPM Hour. Today we have Jill Silver and Lauren Kuehne talking to us about lessons learned from 20 years of management of invasive knotweed. Jill is the director and she's also a watershed ecologist at the 10,000 Year Institute. For those of you who are not familiar with the 10,000 Year Institute, which I also was not familiar with it, so I had to go look it up. This institute was formed in 1992 and works to integrate academic science and applied resource management in decision making. And they partner with a variety of different folks from nonprofits, government agencies, tribes, universities, and also scientific consultants and legal professionals. I did get that directly from the website. Jill received her BS degree from Evergreen State College in Olympia, Washington in environmental studies. And she previously worked as the Invasive Species Program Coordinator with the Kuehne Tribal Nation and as a habitat biologist for the Hoh Tribal Nation. Lauren Kuehne is an environmental consultant at Omnifishient Consulting. She's also a graduate from Evergreen State College with a degree in marine biology and a master's degree in aquatic and fisheries science from the University of Washington. She previously worked as a research scientist at University of Washington in the School of Aquatic and Fisheries Sciences and a research biologist at NOAA at the Northwest Fisheries Science Center. And so today, as I mentioned, they're going to talk about invasive knotweed management on an unregulated Western River. And Jill, I think you're going to lead us off.
Thank you so much, Matt and Steve. Thanks for the support for presenting and characterizing all of this data that we've collected for all these years. So I will Lauren's going to drive and I will start out. So we are presenting 20 years of data of knotweed management on this amazing unregulated migrating river, the Hoh River, on the coast of the Olympic Peninsula in Washington State. And Lauren has been a partner in helping and working with us on pulling our data forward into community and into publication. So we work for Healthy Rivers, and Matt already described what it is we do, and we have been doing applied science for 30 years. We also do a lot of invasive plant management, develop protocols. And the reason that we do this is because it's something that is not well done in the rest of natural resources and watershed management and protection. And we're going to do an introduction and history and cover some of the monitoring and restoration efforts through time. And then Lauren's going to go into analyzing the data and lessons for adaptive management. So for the past 20 years, we've been working on the Hoh River. And the Hoh River is, as I said, this undammed, unregulated river draining the Olympic Mountains from the Hoh Glacier down to the Pacific Ocean. It's a steep river. It's 56 miles long. And the Hoh rainforest is one of the 5 temperate, moist coniferous rainforests in the world. And it gets about 400,000 visitors a year. I've doubled that right now. And we have wild fish on the Hoh River. We have no hatchery on the Hoh River. There is a small hatchery at the mouth of the river on the reservation, the Hoh Indian Reservation, that takes steelhead outplants from the Kuehne River and puts them in the lower river. But other than that, there are no hatchery fish on the Hoh. And the populations are relatively abundant of Chinook, steelhead, coho, bull trout, cutthroat. So we started working on knotweed when I was the Hoh Tribe's habitat biologist. In 1999, we had a 100-year storm event, and this river pulls a lot of sediment out of the mountains, which builds up in the channels, and it constantly avulses back and forth. So at that point, the river avulsed to the south bank into a homestead property. Excuse me 1 second. Crew's calling in. The river avulsed to the south side, and captured a knotweed clump that my stream typing crew had identified the year before in a homestead on the river floodplain, and took a picture and asked me what the plant was, and I didn't know at the time. And a year later, I found it as I went down the river on a float trip with the Bureau of Reclamation, looking at sediment issues on the river. And I found this plant scattered down the river on gravel islands, gravel bars in the middle of the channel. And that began the history of the Hoh River work. So we began treatment in 2001 with funding from the BIA. And those are the size of the plants that we were finding and often captured at the large wood that's distributed across the bars, because we have a lot of big trees that move in the river. And that's a Scotch broom plant behind that. And that's another species that we had to walk by for many years because there wasn't any funding, but we're doing mostly Scotch broom now. Thank you. And from there, we continued to apply for a variety of funding. We got Pacific Coast Salmon Recovery funding for a few years through the Hoh Tribe and National Fish and Wildlife Foundation. And then WSDA started a knotweed program. And that was based on research that Lauren Ergensen did at the University of Washington on knotweed that demonstrated that knotweed has a specific influence on riparian succession, shading out any germination of other species and also changing soil nutrients. And so that's when we also finally, at 2010, we also started treatment for the other species. And that's me getting seeds off of a reed canary grass stem in Elk Creek on the Hoh River. And we're continuing treatment. So from 2010 onward, we had multiple funding. And finally in 2012, we started getting funding through the Salmon Recovery Funding Board, which is the major source of money for restoration on rivers for salmon. And it took Lauren Ergensen's research to prove to the NOAA and NIMF scientists who make the criteria up for what is fundable to make knotweed into a fundable item, and none of the other species were. And so it took until 2015 with the Washington Coast Restoration and Resiliency Initiative, which we helped pull together. It's a state capital budget funded program that is also managed by the State Recreation and Conservation Office, which the Surf Board is also in. And we've been working under that program now since 2015. We're actually getting $1.4 million a biennium to do 26 species in 8 watersheds and 3 counties on the coast, doing a lot of work on other restoration projects and across landscapes. So we just continue doing the knotweed on the Hoh River along with all the other species on the Hoh River at the same time. Thank you. And we do a lot of training. So that's training the Hoh Tribe, and I am sitting at a knotweed plant in the Hoh Reservation floodplain at the mouth of the Hoh River on the left bank. Thank you. And I'm going to turn it over to Lauren at this point to go through everything else.
Great.
Thanks, Lauren.
Thanks, Jill. Okay, I'm just going to sort of change my active view, or there we go. Oh, am I? Okay.
You're on the side. You're good.
I'm good. Okay, excellent. Yeah, so I'm going to pick up where Jill left off. So Jill and I have worked together for many years, informally, formally, and for a long time. Actually, we have talked about the challenges of that. You know, there's often money to monitor or control invasive species, but the funding is often not available to collect, assimilate, synthesize, aggregate, and analyze the data and sort of obtain lessons learned. And so Jill and I finally last year, I knew about this amazing data set that she had been diligently collecting for almost 20 years. And we applied for a grant from WIPM to do the synthesis and aggregation and to try and come up with lessons learned from this very unique and very valuable data set, which I'll talk about more in this next section. This is the first time that I have presented these data, so it's both very exciting, but I may stumble perhaps more than usual, so sure everyone will be very generous with their, except that I cannot seem to move through my next. Oh, there we go. Okay, great. So as Jill said, they have been monitoring and controlling invasive plants. Knotweed started with this, but other invasive plants on the Hoh River for more than 20 years at this point. And I start out with this photo to give a sense of the vastness of that challenge. And we'll talk about that. So the fact that the data has been collected and monitored over this, not only over this time period, but at this large of a scale. This is a very large river. Not only is it a large river, it's an undammed river, and that it's also a steep river that has hydrology of glaciation as well as large floods. And so that makes it a very dynamic river. So we have a lot of movement going on, a lot of movement of water, a lot of movement of sediment. The floodplain is very dynamic, and this makes it very challenging to monitor and capture information over time. So we'll talk about some of those challenges, but I just want to, I think this picture kind of gives you a sense of what that is like. So the monitoring and restoration has effectively gone on. The monitoring started in 2001, but the control work started in 2002. So there is the avulsion point, or the point of introduction to the river, approximately at river mile 30, which is river kilometer 48. And so since 2001 time, knotweed has been surveyed and treated in most of the river reaches in most years. And I'll talk about that. Of course, it can depend on the amount of funding and field funding basically drives how many river reaches were surveyed in most years. But in general, most have been surveyed and treated. The treatment has been over that time period has been consistent. Early treatment years focused on injection using glyphosate of large canes and large plants. However, after a couple of years, the prevalence of large canes really was reduced to the point that it was almost entirely populations of small plants. And so injection is sort of phased out over the first 4 or 5 years, and eventually it becomes basically almost all spray. Glyphosate was initially used, followed by a couple of years of a mixture of glyphosate and imazapyr. And then from about 2012 on, we see mostly imazapyr and then sometimes glyphosate used in isolation for the most part. And these are, for folks that don't know, glyphosate and imazapyr have very comparable control for knotweed, although as Jill has found, the imazapyr seems they're finding better efficacy with imazapyr, but they are both considered some of the best options for controlling knotweed with a single season treatment. This is just a picture of spraying knotweed on the bank. This is kind of a typical area, a forested area. So one of the big survey challenges, I talked about how reaches have been surveyed in most reaches in most years. But one of the challenges of this has to do with funding and crew and availability of field crews in certain years. The habitat diversity, there's gravel bars, there's willow and alder thickets, there's log jams, there are mature forested areas stacked full of native and also other non-native species. So finding these plants in all of these habitats has at times required an intensive grid-based survey approach where technicians are 15 to 30 feet apart from one another, walking across these thickets in order to not miss plants. So this is a big challenge, both finding, especially smaller populations and smaller plants in these diverse habitats, and then also having the field capacity to do these surveys in a way that is robust, that you don't end up missing things. There's also, the log jam is a great example in the willow thickets. Those are places where debris accrues, and so it's very easy for small bits of knotweed to become established in those areas. That's one of the big challenges. Here's a great example. There's a little knotweed plant hiding right in there, naked to the, unless you're kind of right on top of it. Another survey challenge I talked about earlier is that this is an unregulated river. It has a lot of water moving through it from glacier and from it's a rainforest, and they get a lot of rain, especially in the fall. That means that there is a lot of movement of sediments. There's also a lot of new disturbance in floodplains. And so I just chose this section of river to contrast. This is a 13-year difference of the exact same section of river. As you can see, the difference in the later one, that area has become well vegetated, but we have sort of early disturbance in 2006. That would have been a period of some pretty big flooding. So this river is dynamic. It is always changing, and that can be a challenge for both detection and monitoring and control and treatment of invasive plants. So that leads to, we have when we designed this project or designed this work, we had two research objectives. The first was to use this unique, I call it very unique because the fact that these data have been collected and sustained over 2 decades is incredible. It's such a unique data set, not especially in invasive plants. I can't even think of many that have been done this systematically. Mapping has been consistent part of that since the very beginning. Not only mapping of the location of points, but also things like stem classes, size bins, height classes. And so there's actually, for this kind of a long-term data set, the amount of data is really extraordinary. So the first research objective was to use this 20-year data set to look at environmental factors. So we hypothesized that there were environmental factors that would lead to the persistence and areas where knotweed was more likely to establish, and areas where once established, it was more likely to persist even with treatment, because these populations are being treated in every year, right? So we wanted to use this data set in a way to try and identify what are those environmental factors, because if we can identify, is it active channel width, is it grading, is it forested landscape, we can actually use those then to generalize that information and predict establishment and persistence for other rivers, hopefully, at least that's the idea. So not only to understand those factors, but potentially to come up with predictive models that can be generalized to other areas. The second objective was to try and capture 20 plus years of observational data, and which we're calling lesson learned. So over time, this is kind of an adaptive management story, is that as you're treating invasive plants in a new environment, you're learning all the time about what is effective. You're starting to notice things, noticing new challenges. And so there's actually almost 20 years of this kind of information that can be used. We wanted to summarize and synthesize this to say, well, what have we learned? And then what are the important research gaps that are still outstanding that would make treatment and restoration better in the future? Again, trying to generalize these information for other managers in other areas. So this is mostly based on sort of a qualitative assessment and summary of, for the most part, summary reports that were done over that same time period. Okay, so that kind of describes the monitoring and restoration effort through time. This next section is going to be my attempt to tell you the most important parts of the story from these 22 decades of information. And as we were informally chatting earlier, it's a lot of information. And so I have tried to, I won't be able to talk about all of it. And I'm also not going to, get too bogged down in the methods. I'm going to try and get to the part which is the story, the take-home messages. So let's start. So our first research objective, which is looking at the environmental factors of establishment and persistence, right? So just to remind you, the objective is to assess what environmental factors are associated with establishment and persistence of knotweed. Again, we have our point of introduction, which is known at a known time period, 1999. And then, monitoring started in 2001, 2, and then control work started in 2003. So this then becomes our, study area, our monitoring area. And we have what we want to do is we want to model environmental factors that we think will, dictate where is it appearing in the landscape over and over. So we had some variables that we came up with, and we have some hypotheses about the directionality of how those things would be. So proximity to the source. You might expect that proximity to the source, you're going to find bigger populations that are harder to eradicate closer to the source. Now, treatment, these populations are being treated in every year. So we would expect that, the year of treatment or the increase in treatment effort over time would lead to decreases. Active channel width, our hypothesis was that a wider channel gives more room, basically, for knotweed to spread out and more disturbed areas where knotweed can establish. And channel braiding tends to be often related to active channel width, but it is different. So braiding is the number of braids that are within that channel. We thought that channel braiding might have a similar kind of positive impact. Sinuosity is the meander of your river. And again, that was considered, usually indicates a wider floodplain, a slower flow. And so we hypothesized that that would be a positive impact. Percent forested, the idea would be that the forest cover is going to shade out knotweed, or it's going to not establish so well. So we thought that higher levels of forest cover would reduce the prevalence of knotweed. And then prevalence of a wetland, not prevalence, it's a proportion of the landscape that is emergent wetland, or there are different kinds of wetlands. We ended up focusing on emergent wetland because it tends to be flatter, indicator of lower gradient areas. But basically the presence and extent of wetland areas might be an indicator of places where knotweed would do particularly well. So this was sort of there were other variables too that, we've sort of weeded out along the way, but these were sort of the final variables that we said, okay, this is what we'd like to try and capture and compare with our knotweed observations over time. So for an example, here are the points where knotweed was found and treated in 2002. And we have these survey points for each of our survey years. And so we're going to use those against environmental covariates. So the way that we come up with environmental covariates is basically by binning. We're going to create bins. Each river kilometer is going to become a 1 kilometer bin. So our steps, and this map here shows our study area up to about, our study area up to about river kilometer 48. And each of these river kilometers is labeled. So we're going to assign our knotweed points to these river kilometer bins, the nearest river kilometer bin. The next step, which I'll show you in the next slide, is that for each of these bins, we're going to come up with some environmental covariates that what I call characterize this section of the river kilometer. So we're going to characterize the active channel width for river kilometer 1 and 2, et cetera, et cetera. We're going to characterize the percent emergent vegetation in each of these sections of the river, and then we're going to model those. We're going to model the number of knotweed observations against these covariates with year as a factor as well, accounting for the fact that these are collected in separate years. So, for folks that are really enjoy knowing how you come up with environmental covariates using, remote data, here's a description of most of our covariates, what we did. So the two on the left-hand side, active channel width and channel braiding, those are, we take our river kilometer bins and we take 4 samples per river kilometer bin. And actually these are just, you take the points and then you lay them out in Google Earth and then you manually draw them. And then you export that to a shapefile, which then you can bring into your, I use R for most of my data processing, use that as shapefile, that becomes your covariate for each river kilometer. The land cover is done, those are on the right-hand side. So we, again, use our river kilometer points as points, and then we take a 500-meter buffer around these sample points, and we calculate the percent forested, which is from the National Land Cover Database, and then also the percent of wetland. And National Land Cover Database, I'll mention, has other types of land cover. They have different types of wetlands, they have deciduous and non-deciduous, but we a lot of those tend to be correlated. And so the two that we honed in on that were most representative of what we wanted were percent forested and percent emergent wetland. But that's how you capture these environmental covariates. We also did sinuosity, which is just the meander of the river, and that's determined by the flow line. And then it's the straight line between the 2 points on a flow line, divided by the meander. I can't remember the formula. Anyway, sinuosity is another one on there. So So then what we do is we take those environmental variables, and then these become candidate models. And so then you use model selection to say, well, which of these factors should we retain in a best, what is our best model, and what are the factors that actually get retained in that model? And then you run that, use that as your final model. And then we're going to evaluate, excuse me, evaluate the effect sizes out of that final model. So here, our candidate models have all of our variables in there, but when we did the model selection, basically all of the variables are retained in the final model. The only one that is not retained in the final model was active channel width. And that's partly because it's closely correlated with channel braiding. And it also simply, so it's correlated with channel braiding, and it doesn't improve the model very much. So we could retain it legitimately, but it doesn't really offer us too much more information. Okay, so that is the process of the modeling for the environmental covariates. Okay, this is our first result slide, and oh, my animation didn't work. Okay, that's all right. Sorry, it was supposed to animate in, so you get to see all of it all at once. So this is the basic pattern of trends through time, and you kind of have to sit and look at it for a minute to see what you're looking at. So we'll just take a moment and think about this. But this is basically, these knotweed populations are being treated through time, so you would expect them to be reduced. And this top graph here is a heat map or a density plot. This is on a log scale. So the darkest greens are going to be a logged, they're the highest density, the darkest green, all the way up to white, which means that it was surveyed, but it was a 0. There were no knotweed observations in that kilometer of river. So your heat map starts at the top. It starts at river kilometer, it starts at the river mouth, and then going to the right-hand side, it goes up to the higher up to river kilometer 47, 48. And you can see then the density, and then the year is moving up through time. So the way that we would interpret this is that if it's gray, here we go, hopefully you can see my mouse now. So gray, there were reaches that were not surveyed in particular years. And so that is indicated by this gray box here. And so, for example, in 2002, we have no records of river kilometer 0 through 13 being surveyed, so those are grayed out. However, this matches up with our 2002 points here. This I need to investigate, because it may have been assigned a non-surveyed, but you can basically see a correspondence between the high density of knotweed and the high density along the river longitudinally. So then you have to think about it, but it's moving up through time, and we are seeing the density of knotweed disappearing. And there are some patterns where there's, oops, my mouse keeps disappearing. There are some patterns, there are some patterns that indicate that the places that were highest density up in the upriver are still remaining to be the highest density. So highest density here still seems to be highest density up here. And we still have areas where it was low density or zeros, and those sections of river do seem to be kind of lower in density. And so this is where our environmental factors start to come in, is that we might expect that there are environmental covariates associated with these sections of river that are leading to better establishment and persistence through time, which we'll talk about. But this is the general trends, and you can see that, and Jill has a wonderful animation that I decided we didn't have time for, but you can map this over time, and you can watch, the knotweed points disappearing on the landscape with all of that restoration survey, diligent, monitoring work has really, really dramatically reduced these populations. And there are various ways to show this. I landed on this one. So let's look next at, okay, so then this is our model result. I know it looks so exciting, but it is kind of exciting. This is where we're modeling the influence of our different environmental factors on knotweed presence and persistence through time. So what you're seeing here is our variable. You have the river kilometer, which is proximity to source. You have the year, which indicates treatment, sinuosity, channel braiding, forested and emergent wetland. And this is the effect size. So the way that you read this is that each kilometer of river kilometer had a positive effect on knotweed presence. So yes, proximity to source mattered. The year, we're seeing the effect of treatment is declining. It has a negative effect. Sinuosity had a positive effect, so more sinuous, more meandering, we'll tend to find more knotweed. Channel braiding, interesting. Channel braiding was associated with a negative impact. Percent forested, we see a negative impact. So more forested area seems to be associated with lower knotweed presence. And then emergent wetland, this was an interesting one. Emergent wetland had a very large effect. So the presence of emergent wetland is associated with knotweed presence. And so these animations, oops, sorry. So then what I did is these green and red check boxes are the alignment with our hypothesis, right? Is that we predicted that river proximity to the source would matter. We predicted these things. However, this is where our prediction did not line up, channel braiding. And so that's an area where we want to maybe investigate a little bit more. So this is interesting because it does say that, okay, maybe these environmental factors can help us determine where on the river we are going to find knotweed. And again, predict these outcomes out to other areas. However, there's a but. The model doesn't explain a lot of the variance. So our fixed effects, which is our fixed factors, it's only explaining 28% of the variance in the model. And our combined, which means our fixed effects and the random effects, are explaining close to 40%. So it's definitely explaining some of the variance, but it's not explaining a ton of the variance. And so I think that, well, no, I don't think. I know that the next steps are, there is a little bit more to do here. One is that many of these variables are not available in every single year. So for example, active channel width, we only have a couple of time points, but we can go back and make those variables more granular. We can calculate an active channel width probably for almost every year. So those are examples of ways that we might be able to look at improving the model. And then this also will help us investigate further. So why is channel braiding? It's interesting. Why is channel braiding associated with that decline? Is there something else there? Why is emergent wetland associated? Does it have to do with gradient, slope of the river? And so there may be variables that this model can indicate what other variables may either be included or added to it or substituted that will define and predict more of our variation. So, but at least we now have a start. And so then the goal becomes to identify variables that maybe can improve this modeling. This is sort of a bonus thing that I'm working on, which is talking about lateral spread from the main stem. So this is important, is that the distance that the knotweed points are from the main flow line is an indicator of how much knotweed may be spreading and moving on the landscape in response, especially to flood events. And so the lateral distance I've shown in this picture here, these green are, again, the 2002 knotweed points, and the lateral distance is simply the distance to the main flow line, which if the knotweed is spreading, you might expect that if you treat these points in 2002 and you come back in 2003 and they've moved further away from the main stem, that's an indicator of this lateral spread. So what we're hoping is that we can analyze that in a way that we can assess both the rates and the extent of spread in relation to flood events. And down here, so on the Hoh, we're very lucky. There was some research that has been done on discharge events by Amy East in 2017. And they actually calculated the maximum discharge of these events. And they calculated the discharge of different flood events. And so we can actually use that. So I calculated the lateral distance of every single knotweed point in every single year. And so then what you're looking at here, starting at the top, is in 2002, moving all the way down to 2022. So each of these is a year. And what this is the distribution of the lateral distance. So to pick on, say, 2012, this heaping up of the lateral distance, most of these points in 2012 were found close to the main stem. In contrast, we picked 2008, it's much flatter. And so we see a bigger distribution of points that are found further from the main flow line. And what we want to do is relate these to these large, heavy flood year events. And so based on East et al., we can identify some of these big discharge years, which I believe were winter of 2003, and then also it looks like 2006 and 7, I believe, were some big flood years. We don't have discharge data yet for later on, but I believe there was a big one. There's evidence there was a big one in 2019. So when I start to look at the correspondence of the movement of these or change in the lateral distance across these years, there's some indication that it's responding to these often multiple wintertime flood events, but we can do it. I think there's ways to do a better job. I'll show that in the next slide, because one thing we have, oh, sorry, here's the average. So the average lateral distance, for example, in 2002 is 175 meters. It sort of bounces around, but we're sort of seeing, I think, a progression to where the average lateral distance is further away. And I think that what that's an indication of is that there are fewer Knotweed points. So we have to keep in mind that as we're changing through time, the Knotweed number of points of Knotweed is being reduced, and the survey effort does vary from year to year, but I believe that we're seeing a shift toward fewer points that are more spread out. And Jill, I believe we've talked about that. So I think a better way to do this may be on this next slide. What we can do is sort of next steps with this is we can actually contrast specific sites or reaches where survey effort was comparable between the 2 years, pre and post some of these larger floods. And also we can create flow lines that are specific to the year rather than using a single flow line for all the years. So there's some, I think, some really interesting possibilities with looking at this in further. Okay, this is the last slide of this section. The next one we're going to talk about is, it's 12:38. I just want to make sure we have time for questions. So, you know, one of the big results is that obviously Jill and all of the people that have helped have demonstrated that the sustained management, you can reduce Knotweed populations even in a huge dynamic river. So the next steps are to continue monitoring and treating those in conjunction with other invasive species. I think another really important result is that this diligent documentation allows us to analyze and hopefully generalize information for other managers, other scientists, other people that are grappling with how to manage these populations of invasive species. And so for me, one of the next steps is that I think this is an important story and what the importance of documenting and supporting development of data repositories and infrastructure to house these data. And then, you know, the last result is that landscape factors, it does seem like it can explain some variance in the population establishment and persistence. I think we have more work to do in terms of both testing and including additional variables, but then also I would like to use these in trying to continue to refine analysis in order to create predictive models, there are some folks working on that. So I think there's some really interesting next steps in those directions. Okay, the last section, it's 12:40. And yeah, this is a big section, but I'm going to again try and focus on the sort of take-home story. There's a lot here, which is I talked about those annual reports. We're not going to talk about these words. I just want you to see how many words there are for the most part. And this is only a few of the years. So this is, you know, the most boiled down of all of these information that have been, again, diligently documented each year. And we've trying to come up with some ways to standardize these. And this is, you know, an effort in that direction. We have information on acres treated, the treatment, the survey emphasis, changes in survey emphasis, things like, did you do a grid-based survey, or, you know, had to focus, there were, depending on availability of effort, you know, the emphasis had to shift from year to year. Treatment effectiveness, and then also interesting notations on, you know, things that were noted that are important, I think, for this idea of adaptive management. So I've highlighted in red, these are things that when we accumulate all of these in each of these 20 years, what we start to notice is repetition, right? And so in many years, large cane survived only rarely. Population is well distributed in smaller form. Downward trend in size continues, right? There's this repetition of things that we can identify as trends. Those are in red. And then in blue, I've just pulled out some of the interesting pieces of, these are observational data that are repeated in multiple years that can come to trends, and they can also be used to identify research gaps. So I'm going to talk about these in the next slide. So how to capture these, right, is here's our timeline. And when you summarize those information from all of those reports and anecdotal information and observational, well, I don't want to call it anecdotal, it's observational data. We start to see what I consider some themes. Control, so the efficacy of the plants that were treated was typically very high. So the glyphosate and the imazapyr treatments, if you could detect the plants, usually, and the effectiveness monitoring was indicating somewhere in the realm of 95 to 98%, plant death over, and that was consistent through time based on injection and spray. As we've shown, the knotweed biomass, both the number of clusters, the number of stems, and the stem height is all declining through time, which we can show in the data as well. Observationally, there is a repetition of the idea of lateral and upland spread, probably in response to flooding and movement of the river. And so this is and then this is repeated over multiple times, is that then the effort to detect these smaller, fewer plants that are more spread out increases through time. And so these are what I would capture if I had to call, these are the consistent themes that emerge in this summarizing of the information. And so it points toward, I think, the challenge is that you can reduce the plant populations, but then how will you find the reduced populations? And this, I think, is where the environmental analysis comes in, right? Is that our environmental analysis can potentially tell folks where to target their resources, given that they're going to have a harder and harder time potentially finding plants. There's also information that Jill and colleagues have accumulated over time, which is with regard to propagation, seed banks, allelopathy, I can never say that. Anyway, and then also I think interestingly, the time of year, the site access and treatment efficacy comes up many times. So I'm actually going to focus on that, is that there is a window, a treatment window that is very difficult to manage. And it is becoming more and more difficult to manage, is that plants are not emerging in and able to be found until probably August. But then if you wait until September now, they're starting to dry out and from drought stress. And then the drought stressed plants won't uptake the herbicide. And so you can't. So there's the window has been sort of progressively narrowing. You also can't wait too late in the year, because then you get into darkness and clouds and rain and flooding and that kind of thing. So this information on the site type and when plants can be surveyed and treated is a consistent theme that is, I'd say, a consistent and very important theme. The other theme that comes up is really trying to understand the importance and implications of changing climates. So these graphs are actually summaries from that work I mentioned, East et al., which we're really fortunate to have. So what they did is they looked at changes over time from 1940 to 2014, I think. And they looked at active channel width, they looked at braiding index, they looked at discharge, and they noticed changes, consistent changes that could be documented over that time period. The significant widening of all four rivers since the 70s. We see an intensification of regional floods. The strong temporal chill of greater braiding is increased sediment supply due to glacial retreat. So they have actually detected the signal of glacial retreat in the river discharge and sedimentation. Now that gets to the environmental analysis. Is it active channel width or braiding index? And so this ties into our environmental analysis in important ways, is trying to understand the implications of changing climate. Increased flooding, probably going to spread knotweed around more. Increased braiding may reduce establishment. I mean, we kind of have to dive into that more. I think there's a lot that needs to be understood there. And this slide is actually one that Jill had prepared. It's interesting. I prepared this one based on East et al., and she had prepared something comparable. I don't know when this slide was prepared, but talking about how sediment, aggrading sediment causes the large disturbance zones where invasive species do very well. And so we may anticipate and expect more of that in the future with a glacial retreat. And then these were consistent research gaps. So when I summarized all of the information, this observational data over time, these were some consistent research gaps that were identified. And I thought it was interesting because they kind of run, they cross a couple of different, they run from down here, which I call improving treatment outcomes. So herbicide translocation, persistence of the rhizomes. I believe the reports talk about, rhizomes persisting for 8 to 10 years only to resprout. So herbicide translocation is a huge, huge issue, and the resprouting from what you would think were dormant or long buried rhizomes is a consistent theme. The chia pet syndrome is basically huge rhizomes with only very few leaves, and this is a problem for translocation of herbicides. And this has been noted. Hybridization. At first, it started with only Bohemian knotweed, and then later on, giant knotweed, and then looking like hybrids are appearing on the river. Again, the influence of floods and how that is influencing the survey, monitoring, treatment is a consistent theme. And then also the future, the role of glacial retreat and sedimentation. So these are identified as important research gaps that will probably help in the future with, again, continuing to monitor, continuing to survey, continue to do control work. Okay, that's the last slide is I'm sure that this list is probably out of date. I grabbed it from Jill's presentations before, and it's probably
good.
It's okay. Well, there's always more people, but I mean, this is just a huge collaborative project, and there's so many people and organizations and entities that contributed. It's really quite an honor to be able to present the data from their hard work. So, okay,
that's a pleasure to work with you, Lauren.
Thank you. I guess I'm going to stop sharing. Is that, Matt, should I stop screen sharing then?
That's fine. You can stop sharing now if you like. I did notice there was one question in the chat that Jill was kind enough to answer. I don't know if anything else needs to be done there. So if folks want to look at the chat, they can see the one question that was posed so far. Otherwise, if other folks have a question that they'd like to pose, you can either type your, you can either take your mic off mute and ask your question, or if you're feeling shy, you can simply type your question in the chat. And Lauren, I did not think that you stumbled really at all through the talk. So if that was the first time through, it wasn't noticeable. And if you had never said anything, I don't think anybody would have noticed.
I shouldn't have said anything.
You shouldn't have said anything. So I'm not currently seeing any raised hands. We do have, let's see. Well, Bill, do you want to take
I have an answer for that if you want me to just speak it. So
Sure.
Lenora asks about the association of knotweed with emergent wetlands, and might it be the cause or the result? So knotweed does alter the landscape, so it favors its own persistence, but we don't allow that to happen on the Hoh River. So it hasn't been able to do that. But the reason that it would end up in emergent wetlands is the way the river flood flows transport material and how material drops out as they hit obstructions. So the top parts of every meander bend, the side channels that start going through the floodplain with log jams, or the log jams around the bars around the outside of the meander bends, is where knotweed is always dropped out. In other words, the fragments of rhizome and stem that are being transported after having been scoured out, either out of buried sediment or off of a bank, are transported out, and they hit an obstruction and they drop. And you can see the flow of the water just goes over and drops velocity. So that's where you find new plant sites. And I can, at this point, I can anticipate, I can basically identify where we will find knotweed in the following year after big floods. And so emergent wetlands are a place where water has spread way out into the floodplain, and water has slowed going through all of the forest, and it hits the zone of slow, wet site that's ideal for knotweed to grow from.
Yeah, I'm gonna follow up on that. So exactly what Jill said, Lenora, is that I think that the emergent wetland is more of a indicator of low gradient of channel of slow water. I don't think the emergent wetland itself is necessarily the basic habitat. I think it's more an indication, because emergent wetland, where you have that, you have low gradient, you have places where the water can spread out, you have stagnant water. And so I think it's actually really helpful, because it gives us an indication of what environmental covariates might be better representative. So actually channel gradient might be a better indicator. And then if we can capture this discharge, like the flood, you know, some sort of environmental covariate captures what Jill is talking about, where the water basically stops. If we can put that in our model, it probably will improve our model even more. So the emergent wetland sort of gives you a peek into what covariates are better, and then you kind of iteratively try and refine those covariates.
Great.
And I'll answer the next one too, if you want me to, Matt.
Well, I actually think that Bill had a question. He said, I have a question, and I think he's going to, he took his mic off mute and then he put it back on mute as you answered Lenora's question. So Bill, do you want to go ahead and ask your question?
Well, I have a comment. One would be, maybe research needs might be still for some other monitoring. And I think of the drones and the possibility of some surveys in the fall of the year, you know, looking at foliage color or something like that, especially in areas that may be open. You know, the challenge is getting to the site and then surveying the site once you get there. That in itself is a big deal. But the other thing that I was wondering, looking at some of the information, is if flow of the river is a factor, environmental factor. And I just think of where we have the hydro project for Lewis County PUD on the Cowlitz River, where the dam and the water coming into that backwater of the impoundment, you know, slows the water down, and you know, that knotweed isn't establishing on those banks like it does, you know, a mile upstream or so from those impoundments. And so I don't know whether that's the same thing that's happening in the lower part of the Hoh. But anyway, I was just kind of curious about that. But it also maybe factors into what you was just describing with these wide meanders and where plant material is dropping out. So.
Yeah, I think like sinuosity and braiding get at that a bit, because of course they're related to flood discharge. And again, I think like this initial modeling kind of gives us a peek into saying, okay, well, sinuosity is clearly meandering channels are an indicator. So that is an indicator, you know, there's an association with flow, stoppage. It's kind of more, I think at this point is more a matter of fine tuning it and saying, well, what is the covariate that, or what the covariate or the suite of covariates that are going to best sort of capture these phenomenon. So yeah, but I think flow is clearly indicated as you know, being important. The other thing that I don't know if I talked about was we did these in 1 kilometer bins. It is probably possible, well, it's not possible, I know it's possible. We could try a model that is a finer scale. We could do 0.5 kilometer bins or something like that, right? It's possible that fine tuning it would it be running it at a different scale of covariates.
Yeah. And Jill has typed in an answer to the question about the drones. She writes, drones are a tool that we use regularly for species like Scotch broom, but for knotweed, we have so little and so much is under canopy that it's not helpful there. It is helpful in most other river systems. Let's see. So, Jill, did you want to take a stab at Len Coop's question about the Japanese knotweed psyllid?
Sure. We've participated in all of the efforts over the last 20 years to develop biocontrols for knotweed, but we really have so little Japanese knotweed or any knotweed on the Hoh River anymore that it's not enough population to distribute to. Otherwise, we would have. We did just distribute Scotch broom beetles, bruschettus, in 3 watersheds, and we probably will be distributing the psyllid in perhaps the Quileute, the Hoko, the Queets, and the Quinault, or suggesting it, but we don't know that yet, whether it's helpful. Thanks for the question. And it's only, biocontrols are only one part of integrated vegetation management. And you have to, in rivers like ours that flood so high that species, end up underwater. Of course, knotweed is usually gone by the time the floods happen. It's usually down to the ground. So I don't know. Anyway, it'll be interesting to watch it in the next decade.
Great. Let's see, I'm not seeing any other questions currently. I just have a quick comment. It's not so much a question, but this certainly points to something that we talk about quite a bit, and that is that local efforts to control weeds are so immensely important in terms of achieving success. And this is a very clear example of that, where you've got a local group of people who are passionate about getting this work done, and it gets done. And without those local efforts, these things just don't really, we don't have great success. So local leadership is absolutely critical, and I think this is a perfect example of that.
Appreciate it.
All right, I think that you guys are off the hook. We don't have any more questions. Let's see, I'm just going to announce that we do have an August 9th IPM Hour. That's going to be Kristen Bowers from New Mexico State University, and she's going to talk about puncture vine, which is near and dear to all of our hearts here, where at least those of us who ride our bicycles to and from work or anywhere else. So she's going to talk about that. So I hope you all can join us for that. Steve, are there any other announcements?
No other announcements. Thank you, Lauren and Jill. That was wonderful.
Thank you again for joining.